Okay, uh, testing, video check. Okay, so a pleasant good night, everyone. Good night, good day, depending on what time, of course, that you are viewing your assignment. So today we'll be looking at the blood. Blood in humans, something that is very important. So we'll be looking at it and we'll be speaking a little bit towards its function. Now, blood is very critical in maintaining homeostasis within the human environment. Homeostasis, as mentioned previously, that's the maintenance of a constant internal environment, and blood is critical to that. It is classed as a connective tissue because it connects in terms of circulation. It really connects everything within the body, and it accounts for 8% of the total body weight. That is very important as well to note. So, in terms of this presentation, the first thing we want to look at is the functions of the blood. It could be classified under three things: transportation, regulation, and protection. It transports gases via the presence of the protein hemoglobin. It transports oxygen, and as well, it does transport carbon dioxide in solution. It moves waste products because of the fact you could view blood as a all, it carries everything within the body itself. So not only does it carry important oxygen and nutrients needed for not only homeostasis, but to maintain metabolism within the body as well. But what it also does, it removes waste, and that is also critical. In terms of regulation, in terms of the functionality of the blood, regulates pH, fluid balance, and heat. Fluid balance is critical in terms of the inter and intra cellular environments. It's very important in terms of those fluid balances. pH, it does regulate pH by its very nature, and of course, carrying of heat, carries and distribute heat throughout the entire body. The last thing, protection against disease, that is most critical. And we'll be looking more at that in terms of the function of the blood when you look not only at the red cells, but also at the white blood cells, the leukocytes. And we will speak a little bit more to that as well when we do examine the lymphatic system, which is one of the repositories, the main repository for those white blood cells, B cells and T cells in particular. All right. And then blood loss, prevention of blood loss, it does that by formation of clots, which is critical in stopping blood loss. Of course, the mechanism regulating this clot formation is one that is under the control of a positive feedback. What does that mean? Well, the more blood that is lost is the more the body tries to form clots to stop its loss. That's a positive feedback mechanism. Another positive feedback mechanism, which we have already encountered and mentioned, is of course oxytocin, critical for the delivery process. More oxytocin that is released into the blood, more contractions, uterine contractions occur to squeeze that bundle of joy out into the external environment. Okay. So let's speak to some of the constituents. So the liquid portion of blood is plasma, and it also contains formed elements, and these refer to the cellular portions. So if you have a look at whole blood, you would notice it consists about 55% of plasma, 45% of these formed elements. And what are these formed elements? Nothing more than red blood cells or erythrocytes, which is forms 99.1% of the formed elements, leukocytes and platelets. White blood cells, leukocytes, very important to fight infection, platelets, critical in the formation of plugs or clots, really critical in formation. And we see that when we look at that clotting cascade which exists, which occurs. The plasma, which is the liquid portion, about 1%, contains a set of other substances, but it's mainly water and proteins. And one of the major proteins present in the blood is that maxitaxia of proteins, that big carrier, albumin, major protein found in blood, which is why, if you would think about it, albumin is also found in eggs, the white of the egg. And this is one of the reasons why when you do smell fresh blood, it does have a smell that is very similar to eggs. This is, of course, due to the presence of albumin. So right now here we're just looking at a smear of a red blood cell smear, and what we're noticing, as seen um, in this smear, we see the platelets. Platelets are really small fragments, critical, as I mentioned, in terms of the formation of clots. The leukocytes, white blood cells, erythrocytes, biconcave discs. We'll speak more to those in just a minute. So the blood plasma components, mainly water and protein. As I mentioned, the major protein is albumin. Albumin is nothing more than a carrier protein. So it could be likened like a maxi or a bus. And in terms of traveling or transporting lots of things, you know, and when we think about it as well, you could think of the blood or the cardiovascular system that moves blood away from the heart throughout the body via the veins and arteries. You could consider all those veins and arteries like the roadway system present, let's say here in Trinidad, and the maxis and the buses, they're carrying these proteins, they're carrier proteins. So they stop along the way, they pick up proteins and they carry them from point A to point B and drop them off. Clotting factors, we mentioned those. Antibodies, very important in terms of the immune system. Antibodies are produced in response to the presence of antigens. And then we have the complement system, which we'll speak about a little bit more. So, the formed elements, red blood cells, erythrocytes, they're producing red bone marrow from hematopoietic cells, specifically hematopoietic or red blood cells initiators. The ones that make them are known as not only hematopoietic, but specifically hematopoietic stem cells, very important in the manufacture of the red blood cells. The white blood cells, they consist of neutrophils, eosinophils, basophils, lymphocytes, monocytes. And as you mentioned, platelets, these are the fragments critical for formation of clots. Let's have a look at these red blood cells. The red blood cells are most numerous and they're biconcave, and the mature cells are anuclear. Now, anuclear means it doesn't have a nucleus. Why is that so? Well, they are so efficient due to the presence of hemoglobin, which is a protein, an oxygen hungry protein, as it were, in such that the red blood cell could do its functioning without really a brain. So what does it do in order to maximize the carrying capacity of those cells? It kicks out the nucleus when it matures. So it kicks out the nucleus when it has reached maturity, such that it could maximally carry oxygen. And of course, the reason why it could carry oxygen without really much quote unquote thought from the brain or the nucleus is because of the fact the presence of hemoglobin. You can think of hemoglobin like an oxygen magnet. So once it sees oxygen, it sucks it in, right? And it takes it and it is now carrying it along. Doesn't need any brain to tell it, hey, look, there's an oxygen, pick it up. No, 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 no. It does it instinctively. Okay. So the hemoglobin binds the oxygen and it carries hydrogen ions, which is also important for the buffering system. These hydrogen ions and maintenance of that pH in the blood. It also carries CO2 for elimination. You speak more to that when you look at respiration, but very simply, one of the things you do have to remember is that this carrying and release of oxygen and CO2, this is done down concentration gradients, and this is done at the level of the alveoli within the lungs itself, but we would speak a little bit more to that when you look at respiration. Here are red blood cells seen under a scanning electron microscope. Note their biconcave discs, right? They have this concave shape on either side, and again they're maximally uh, sh- structured to carry oxygen. So you can think of the heme protein present in here, and it always has iron associated with it, which is why when you do have an iron deficiency, it does affect the carrying capacity of oxygen in your blood, and therefore persons who are anemic, they tend to be weak because of the fact not enough oxygen getting to the cells to break down that food to produce energy in the form of ATP, right, which will have you nice and sprightly. So because of that lack of oxygen, yes, and by extension, decrease in formation of ATP, that's why you have this weakness associated with anemia. All right, so the white blood cells, right? The leukocytes, they're colorless, round, with prominent nuclei. And that's how you could tell them apart because of their structure. They have different nuclei formation. The granulocytes, so-called, because they are grainy, and the agranulocytes, they don't have grains with them. And here, two of them, mature neutrophil and a band-celled. And you could see there are differences in the nuclei, and that's how it could be um, tell the difference between one and the other. Some have phagocytotic activity, and what does that mean? That means they could ingest materials, foreign materials or microorganisms, and destroy them by enclosing them in a phagocytotic vesicle. Then, of course, what happens after it is encased in this vesicle, lysosomes then fuse with the vesicle, or in this case, is taken in, and then they release their lytic or cutting up enzymes. And of course, when they release them, this leads to the destruction of the microorganism, and then the waste product or residue is egested or sent out from the uh, white blood cell that is doing this phagocytotic activity. Very important. All right. So the constituents, the smallest ones, we mentioned that platelets, and the major form or the major reason behind having these platelets is that it is essential for blood coagulation. Here again, we are seeing some erythrocytes, red blood cells, and here we see the platelets. Okay, so now we're talking about hemostasis, not homeostasis, but hemostasis and coagulation. Hemostasis refers to clot formation. It prevents blood loss when blood vessels rupture, and events include contraction of smooth muscle, platelet plug formation, and the clot. Now, this is done via the cascade. There's a cascade involved, and it leads to the formation of prothrombinase. You could tell by the ASC that's an enzyme, and this prothrombinase leads to the conversion of prothrombin into thrombin, and then, of course, conversion of fibrinogen into fibrin via the thrombin. So you have this cascade in terms of an injury to the vessel wall. This initiates this cascade, and of course, leading to the conversion of prothrombinase into prothrombin. Prothrombin then catalyzes the reaction, leading to the formation of thrombin, and then thrombin causes fibrinogen to form these fibrin threads. And these fibrin threads are like a mesh, and they encase the red blood cells, leading to the formation of the clot itself. So this cascade is very important. It is, of course, initiated when there is a tear or some rupture in this vessel wall. This initiates this cascade, right? Very important. Conversion of prothrombase into prothrombin. Prothrombin then, of course, um, under the is converted into thrombin. You under the activity of calcium, prothrombin is converted into thrombin, and then thrombin acts as an enzyme, leading to the conversion of fibrinogen into these fibrin threads. And these fibrin threads, they then actually act like a mesh. You see them here, looking like ramen noodles, right? These white threads, and what their function is that they encase or they encapsulate these red blood cells, leading to the formation of a clot. Very important. All right, so blood types. All right, so blood types must be compatible. This is very important. And there are certain proteins on the outside that, that ensures that this could happen. Right, and this chart here speaks to the blood types A, B, and O, and in terms of who they can donate to. There's a very nice link in the e-classroom which actually shows this relationship. So I would direct you to there in terms of picking it up. Now, in terms of the blood typing, this shows us that how blood typing is done, because as we mentioned, it's very important that you do have the correct type of blood in terms of giving someone a transfusion. Giving someone the wrong type of blood could put them into hemolytic shock, right? So that is something that you want to avoid as a professional. And so to do that, you have to get the right blood type. And this just shows how the typing process is done. All right. So the blood type, right? Red cell antigen, it does speak to it in terms of rhesus positive and rhesus negative bloods. Now, one of the things in terms of this rhesus incompatibility, I just want to highlight, and this will be treated in a little more detail when you're looking at reproduction in terms of the delivery process. If during the first pregnancy you have a rhesus negative mother and a rhesus positive baby or fetus, now what happens, of course, the placenta prevents, of course, the exchange of blood between developing fetus and mother. Sometimes, however, you could have the mixing of blood occurring due to rupturing of some of mother's capillary, which could leak into the fetal environment. So the fetus will be exposed to the mother's rhesus factor, and what it does, it builds up antibodies to it. All right. Now the thing is, at the second during the second pregnancy, now there are what would happen is that there will be those antibodies present, circulating antibodies present in terms of the mother actually having those uh, antibodies to attack the rhesus positive proteins. So if your mother has a second child now and there is rhesus positive as well, those antibodies which are present in the mother's circulating blood, what will happen? They could cross the barrier, get into the fetal circulation because, as I mentioned, through capillary rupture or some type of trauma, you could have the mixing of the blood, and what that leads to, of course, that's a problem because now in terms of the mother's antibodies is recognizing the fetal blood as something that is alien and begins to attack it. All right, so this is hemolytic disease of the newborn, and again, this only happens in during the second fetal during, during the presence, the second pregnancy of a rhesus negative mother who had a previously rhesus positive child. Exchange of blood led to the formation of antibodies in the mother's circulation. Second child, those antibodies crossing that blood barrier, the placenta, through some type of rupture of capillaries or some trauma experience and getting to the fetal circulation will begin to attack the fetus, which is why it's very critical to go to clinic and also to be aware of the fact that if previously you were in this state in terms of you being rhesus negative, child being rhesus positive, that you will be on the lookout that this is an eventuality. There is medication, sorry, which could be taken that eliminates this eventuality. All right, so blood components. So blood is stored in blood banks, right? And it is donated before elective surgery, and you will see this in your professional career. In terms of whole blood transfusion, this is only given for loss of a large volume of blood, massive hemorrhaging, and during or after an operation. Those are the major things you need to know for this class. All right. So use of plasma, right, is used to replace blood volume, and it is used to treat circulatory shock, circulatory failure, which could lead to shock. And that is all the things we need to know concerning that aspect. All right, so blood disorders or abnormalities. Anemia, a low level of hemoglobin or red cells. Leukemia, which is an increase in white cells. And clotting disorder, an abnormal tendency to bleed. So let's talk about anemia. You have hemorrhagic, hemolytic, and sickle cell anemia. Sickle cell, so called because of the sickling of the red blood cell. So, of course, this leads to two problems: one, a decrease in the carrying capacity of oxygen, and an increased likelihood that because of its shape, these blood cells could lead to the formation, they could stick to the inside of capillaries, to blood vessels, and lead to severe pain for the afflicted. All right, so this is just a blood smear showing sickle cell anemia. Notice characteristic sickle shape And as mentioned, the sickle, because of its shape as opposed to a normal red blood cell, which is round, this could lead to sticking within capillaries and blood, different blood vessels, which is exceedingly painful for the person who has the disease. Leukemia, right, characterized by enormous increase in white cells, and of course this abnormal growth, often referred to as cancer of the white blood cells. They could be seen or they are noticed in terms of a blood smear. You see a higher than usual amount of these cells. Right, clotting disorder. For our purpose, we just have to appreciate the clotting cascade, and do know that clotting disorders could occur when this cascade does not function as it should. All right, so blood studies in terms of hematocrit levels. So per 100 milliliters of whole blood, the range, adult range for men is 42, for the hematocrit 42 to 54, and for the women it is slightly less, 36 to 46. Let's have a look. So here you see the in terms of the hematocrit, the plasma, white cells and red. This is the normal that is shown here. One of the things we will notice for anemia, of course, the red blood cell count is down significantly when compared to the normal. And for polycythemia, we notice an elevated amount of red blood cell, decreasing plasma. All right, so these are some of the markers which are used in terms of looking for the normal as associated with blood. So grams of hemoglobin per 100 milliliters for adult men is 14 to 17, whereas for women it's in the 12 to 15 range. In terms of blood count, the range is 4.5 to 5.5 million per microliter, and the white is 5 to 10,000 per microliter. And the range for platelets are 150 to 450,000 per microliter, and these are the normal ranges. Blood slides, the smear is very important to look to see the red cells, and of course to look for platelets. And again, sometimes invaders which might have eluded or escaped the attention of the immune system, such as parasites, it could be found through viewing a blood smear. All right. So in terms of the chemistry, some of the chemistry tests, these are normal ones in terms of when you do send the blood sample to the labs, there are normal ranges associated with those. All right. These are not on the syllabus, and we'd stop there for today. All right, so what did we look at today? We looked at the composition of blood, and mainly we did speak to the major carrier component associated with blood. That's the red blood cell, and of course, it does possess that very critical protein hemoglobin, which allows it to carry oxygen. Hemoglobin, nothing more than an oxygen magnet. Sorry, it takes up oxygen in great quantities and moves it through the blood. Again, we also looked at the concentration, different concentration of the different substances within the blood in terms of protein content, in terms of plasma, water content as well. And we look at the variations that occur. So those were some of the important things we looked at. So that's the end of the lecture for blood. I do hope you enjoyed it, and I will see you, or you will be hearing from me again in the subsequent lectures. All right, good day. Hope you enjoyed it.